Hello everyone, this is SNEH PURBIA from group 316. Here is my video for the topic by physics of the vision. So let's start this video. Visual coding and the retinal receptors. We know each of our senses has a specialized receptor that are sensitive to a particular kind of energy. Receptors for the visions are sensitive to the lights. So let's see. Receptor uh, transduces converts energy into electrochemical patterns. Law of specific nerves energy states that activity by a particular nerve always conveys the same type of information to the brain. Example, that is impulses in a one neuron that indicate light, impulses in another neuron indicate sound. The brain does not duplicate what we see. The sensory coding is determined by which neurons are active. Light enters in the eye through an opening in center known as pupil. Light is focused by a lens and cornea into the rear surface of the eye known as retina. The retina is lined with the visible receptor. You see in the diagram. So let's see about the diagram of an eye. See, human eye. We know we have a layers of a eye. First, the outer layer, which is made up of sclera and cornea, that is a fibrous tunica. We know its name is a fibrous and a vascular. The middle layer is the choroid layer, which is a vascular tunica, and the inner layer is a photosensory tunica that we know is the retina. In the middle layer, we have vascular tunica, vascular tunica, eternal, and the posterior. Let's understand the diagram of the human eye. So, this is the cornea which I am talking about. This is the pupil, the place behind the pupil we have lens, and behind the lens we have vitreous chamber where the vitreous humor, that is the uh, gray jelly like structure which is present inside the eyeball, which can maintain the shape of the eyeball. And on the back side we see the photosensitive cell, that is the retina. Here in the retina, we have in a fovea centralis where only the rods, only the cones are present. We have a blind spot where there is no photosensitive pigment are present. These fovea central is also known as a yellow spot or macula lutea that contains rods and cones, and maximum quantity of cones are present here. Then we have a iris here. We have a ciliary body which attach there. We have a suspensory ligament which can attach lens to the ciliary bodies. Uh, in we have a aqueous chamber here. So let's talk about the retina. It is a photosensory layer. It having many layers. Mainly we can talk about the 10 distinct layers of the retina. First is the pigmented epithelium, second is the rods and cones layer, third is the outer limiting membrane, fourth is the outer nuclear layer, fifth is the outer flagiform layer, sixth is the inner nuclear layer, seventh is the inner pleriform layer, eighth is the ganglion cell layer, 9 is the optic nerve fiber layer, and 10th is the inner limiting layer. These layers will help to vision more precisely. Now let's study the steps of the vision in a light and a dark. First, step 1. Light enters the eye through the cornea. Pupil adjust in response to the light. The lens focuses the light into the retina. The light is focused throughout retina with contained nerves known as a photoreceptors, rods and cones, responsible for the transformation the light rays into an electrical impulse. Fifth is the optic nerve transmit the visual information to the brain, that is the visual cortex. And sixth is the perception of the visual response will occur. Now let's see the visual cycle here. When the light fall on the rhodopsin protein, it will activate and the process is started of visual cycle. This rhodopsin will convert into a transretinal that will convert into a transretinol. This transretinol will convert into a transrethyl ester and which convert into a cis retinol, and this cis retinol convert into cis retinol, and this will again convert into a rhodopsin. And this same thing occur in the dark adaptation, but in the dark adaptation the rods have protein called as rhodopsin protein. This rhodopsin protein will which inactivate by when the light enters the excretion of excitation of the nerves will occur. All transretinal will activate and release of the opsin protein will occur. This will cause retinal isomerase and 11 cis retinal will form, and addition of the opsin protein will again form the rhodopsin protein. So let's understand this more accurately. First, in an bright vision, so what happened, the rhodopsin protein will activate, which will cause the sodium channel closed. So the roads will hyperpolarized and no glutamate release will occur to the polar cells, so that the bipolar cells is dipolarized. So this will occur in a bright vision, but in the dark adaptation, in the dark light option, where in a dim light what will happen, the rhodopsin protein will inactivate, so that the sodium channel will open and the rods become depolarized. Due to this the glutamate release will occur and the bipolar cells will hyperpolarize, so that the process is occurring. So let's understand the process, the mechanism of the vision. So first the incident light will fall, then the structural changes in the retinal or a photopigment will occur, transformation changes of photopigment will occur, so that the activation of the transduction will occur. This activation of transduction will cause the activation of the phosphodiesterase, which will reduce the intracellular cyclic GMP, so that the closure of the sodium channels will occur, so that the hyperpolarization will occur. That will cause the decreased release of the synaptic transmitter, that is the glutamate, which responds in a bipolar cells and other nuclear elements, that will cause the hyperpolarization inhibition. You can see in this diagram also, this is for the day, this is for the night. In a day you see the hyperpolarization of the rods will occur, but in the night the depolarization of the rods will occur. But in the day the depolarization of the bipolar cell will occur, and then in night the hyperpolarization of bipolar cell will occur. This is the difference in the adaptation of dark and light. Let's talk this on the diagrammatic way. See, this is the mechanism of vision, binocular vision, in a diagrammatic way. Scotopic vision in a dim light, rhodopsin pigment we have. So when the opsin protein and the retinal, cis retinal will bind in the dim light, the bleaching will occur. In dim light the bleaching will occur. So in that case, this cis retinal will convert into a trans retinal. When this happens, the opening of a sodium channel will occur and the depolarization cause, so that the optic nerve send this impulse to the occipital nerve. But uh, in bright and dark light, hyperpolarization will occur and this cycle will continues. Now let's talk some basic properties of a light. So, we know that the visible electromagnetic radiation that is represented in the lambda is the 380 to 760 nanometer. Shortest wavelength we know is the UV light, and the longest wavelength we know that is the IR or ionizing radiation, and the visible light source. Medium in which the light propagate is called as optic medium. Light, that is the visual light source, are the natural like sun, or a man-made that will be artificial, that is like light bulb or fluorescent tube, etc. See, a polychromatic or a white light consists of a light of variety of the wavelength. Monochromatic light is consist of a single wavelength. According to the phases, characters of a light can be coherent or incoherent. Coherent lights are the light wave in phase, means one another, that is they have some phases in the same distance from the source. Incident light are the light waves that are not in phases one another. Example, light from a light bulb or the sun. Uh, let's talk about the process that is known as reflection. Law of reflection is the angle of reflection is equal to angle of incidence. Refraction, it is a property when the light passes from one medium into another, the beam changes direction at boundary between two media. We have a formula that is called as n is equal to c by v, that is a dimensionless. Here n is the index of reflection, c is the speed of light, and v is the speed of light. We have different formulas also, that is the Snell's law, Lensmaker equations, that we can talk in later videos. Okay, thank you for today.